Okay, I think we'll get started. Please feel free to get food leisurely and while you're listening, and go back and get more food, I guess, if you need it as you're listening. But we can proceed with the proceedings while you're doing that. Those of you who saw an early agenda may think that I'm Bernard Nalen, the Deputy Coordinator for the President's Malaria Initiative. I'm not Bernard Nalen. My name is Carter Diggs. Bernard had a conflict that he just couldn't deal with in terms of the decision and couldn't make it. And he asked me to fill in, which I'm very happy to do and be a part of this celebration. I am involved with the USAID Malaria Vaccine Development Program, and as such, very, very comfortable in this building. I have many colleagues. We work closely with MVI in a very fruitful relationship over a number of years now, and moving towards that aspect of the solution to the problem, if you will. Of course, and I could speak with you ad nauseum, about the Malaria Vaccine Development Program. I'm not going to do that. That's not what this is about only. We're here to look at the problem of malaria writ large, and in particular to celebrate past contributions to solutions to the problem. So I think that everyone here is aware of the rather dramatic progress that's been made in the field in terms of mitigation of morbidity and mortality in the last 5, 6, 7 years. And that's due to the work of a lot of people. The PMI certainly started in that time frame, the President's Malaria Initiative. If Bernard were here, he could tell you a lot more than I can about it. Linda Banda is here and with the PMI. Where are you, Linda? Yep, raise your hand. So Linda can talk to you at length about the PMI as well during the discussion period or later. What I will say about the PMI is that this approach is a matter of working with countries, 17 countries I believe now, to scale up known interventions to achieve significant impact. And I'm talking about, the usual suspects, impregnated nets, indoor residual spraying, and rapid diagnosis, and artemisinin combination therapy. So the major progress has been made, but I think many people in and outside of the endeavor are worried now about the future, in the sense that if you relax your vigilance, which is likely to happen when funds diminish, or when it looks like you're succeeding, that's exactly when the parasite will bite back with a vengeance. So what we really need to do is to try to be smarter than the parasite. So far we've failed to do that, but that's what is called for here, to try to learn the parasite's tricks and circumvent them. And that's what PATH is trying to do in many ways. With MSEPA, in which they are looking at what's happening on the ground, surveillance, trying to catch the parasite doing its tricks, and then design appropriate strategies to circumvent those tricks. And through MVI, trying to produce new modalities, vaccines that can be added to malaria control programs. And then through their new affiliate, OneWorld Health, doing some really amazing things with technology, new technology to develop drugs. Another source of artemisinin, for example. So today we will be very fortunate to hear 3 distinguished speakers. Hugh Chang is the CEO of OneWorld Health on the screen. Good morning, Hugh. Oh, good. Yes, I guess.
Morning.
It's almost afternoon for you in Seattle. It is afternoon, I guess, yeah.
Actually, we're the other way.
Oh, okay. Well, good. You're right. I always make that mistake. Yeah, right. So my condolences for the hour. Yeah, right. Yeah.
I'm happy to be here.
Rick Steketee with MSEPA, and David Kaslow, the new director of MVI. And I want to say that Well, David and I go back a long ways. We, as they say, David's most recent position before joining MVI was Vice President of Vaccines and Infectious Diseases at Merck, where he learned the craft of vaccine development to a fine level and is bringing that expertise to MVI. But he got his obsession some years ago at NIH and hadn't been able to shake it off. And so I think in his own words, he said that it's unfinished business that he hadn't taken care of, and now he wants to take care of it. So David, the floor is yours. Look forward to hearing from you. Really glad you're here.
Thank you very much, Carter. I really appreciate it. And good morning and welcome to PATH. Now, I would like to take a minute or two this morning just to tell you how great it is to be back in Washington, D.C., and to be back working 24/7 on. It's on? A little closer. There you go. So, and also it's indeed an honor actually to be joining the team here at the PATH Malaria Vaccine Initiative at a really interesting point in MVI's history. Having served 3 years as on MVI's External Advisory Committee, I've had the opportunity, actually have taken the advantage of being able to watch development here one step removed and to really see what MVI has brought visibly and publicly to the efforts of developing a malaria vaccine, but also to see what's completely behind the scenes sometimes in terms of vaccine development. And there are 3 things that I'd like to share with you this morning that I hope will help you see how MVI and the program here at PATH, as I've come to see it both as an external advisor and then as the new director, of actually moving things forward in terms of malaria vaccine development. And the first is that MVI is all about partners. Next week, I hope some of you will be able to join us on Capitol Hill for the Malaria R&D Expo. And there you can meet some of MVI's partners from outside the Beltway, Johns Hopkins, University of Maryland, Seattle Biomed, Inovio Pharmaceuticals, and Emory University, just to name a few of the more than 50 partners that we work with here around the world at MVI. But here today, however, I think it's important that MVI acknowledge our partners in the U.S. government, whether it's at USAID, the National Institutes of Health, the Department of Defense, the Centers for Disease Control and Prevention, or the FDA. And to take a historical perspective, malaria and the U.S. government go way back, actually, to the founding of this country, because one of the first military expenditures of the Continental Congress was $300 to buy quinine for George Washington's troops. So fast forward about 200 years later, in the mid-1980s, it was researchers at Walter Reed Army Institute of Research, or RARE, that partnered with researchers then at SmithKline Beecham in the development of an investigational vaccine, which we now call RTSS. And today, RTSS, this candidate, is under development by GlaxoSmithKline in partnership with MVI, and is actually in the very late stages of development in Africa, across Africa. And of the 11 sites that are involved in this very large-scale safety and efficacy trial, 3 are in Kenya, 2 of which are run with U.S. government partners, one with Walter Reed and the other with CDC. Back here in the United States, MVI has partnered with the U.S. military malaria vaccine program, whether through RARE or the Navy Medical Research Center, in over 6 projects over the last 2 years. And as I mentioned, the CDC is the northern partner to one of the RTSS trial sites in Kenya, all of which are part of the Kemri Medical Research Institute. And there in Kisumu, Kenya, African scientists are in key leadership roles in keeping with CDC's mission to create the expertise, information, and tools to protect people's health. And this is a vital aspect of CDC's work and has made the CDC a source of malaria expertise around the world, be it the head of the WHO's global malaria program, which came from the CDC, or my colleague here today, Rick Steketee. So equally important is the CDC's role as the implementing agency for the President's Malaria Vaccine Initiative that ensures that bed nets, drugs, rapid diagnostics, as perhaps one day, hopefully, a vaccine, can reach the right people in the right place as quickly as possible. Like the U.S. military, the National Institutes of Health are also a constant source of life-saving health research and innovation, and MVI's relationship with the National Institutes of Allergy and Infectious Diseases at NIH are long-standing and deep. Today, MVI is partnering with the Laboratory of Malaria Immunology and Vaccinology at NIAID in a pivotal first-in-human trial of a transmission-blocking vaccine approach, which I'll come back to at the end. We're also partnering with the Laboratory of Malaria and Vector Research in developing and running standardized assays to support many of our vaccine feasibility and translational research projects. Last but not least, our partners at USAID, which has made possible important work on the blood stage vaccine approaches, filling a critical gap in the funding landscape and helping to ensure that we don't close off any options or shut any doors in our search for an effective malaria vaccine. USAID has also supported crucial process development and manufacturing work in another vaccine project undertaken by MVI. This is one done in partnership with the vaccine division of Johnson & Johnson. And then that project had moved into the clinic with support by the Bill and Melinda Gates Foundation. And this particular project is just another of MVI's drive to next generation vaccines, even as we work to do the groundwork for the world's first malaria vaccine, hopefully RTSS. So the power of partnering with the public sector is critical, and malaria vaccine research simply would not be where it is today without the contributions of the U.S. government over many, many years. And what makes history so exciting, however, is the way that MVI has been able to bring diverse public and private sector partners together, and as a result to deliver on our mission of accelerating vaccine development. And MVI is privileged to work with all of them either as partners, as collaborators, as co-investigators, and as scientists. Which brings me to my second point, and that is that MVI is all about the science. Since MVI's beginning at PATH in 1999, MVI has rigorously assessed dozens and dozens of projects for their scientific merit at the outset, then assessed again when specific milestones are reached according to agreed upon criteria. Outside experts are brought in on projects on a regular basis to assure that we are making the best data-driven decisions. And based on these objective assessments of data, MVI has had actually to make the very hard but data-driven decisions to kill projects that don't have the legs to go the distance to either regulatory approval and or feasible implementation. Which leads me to that the science actually has to work in the field. And so the other half of MVI's mission is about availability and access. This means that a vaccine like RTSS not only has to show impact in reducing disease, but it also has to be shown that it can be delivered in a way that works for the kids and their parents, that works in the country and in the local healthcare system, and that it works with existing control measures, as Rick will talk to you about. And my third and final point is that MVI is firmly focused on the future while learning from the past. So every project, whether it's killed or it moves to the next stage of development, contributes to our knowledge base. MVI works across a whole portfolio of projects so that we can quickly apply the lessons learned from one to another project, and perhaps most importantly, hone in and ask the right product development as well as scientific questions. So what comes next? At the end of this year, a second set of results are scheduled to be available on RTSS from the large phase 3 trial ongoing in Africa. And these results should tell us how much RTSS reduces malaria illnesses in infants, the primary age group for whom this vaccine is currently being developed. And we're keen to learn whether or not the data in infants will confirm what we saw in the older kids last year, which is a roughly 50% reduction in malaria cases among those receiving the vaccine. We anticipate that we'll have all the results at the end of 2014, following which our partner GSK will be able to submit a full data package to the European equivalent of the US FDA and to the World Health Organization. In the meantime, GSK and MVI are planning a post-approval program that includes studies to track vaccine safety on an even larger scale than that which was done in the phase 3 trial, and to better understand how RTSS will work in real-life vaccination programs. And then looking further ahead to that much longer-term goal of malaria elimination and hopefully eradication, we have to think critically about the next generation vaccines or vaccines that will need to be brought to bear along with other malaria interventions. We live in a changing world, and the goals that were set a decade ago for malaria vaccines may no longer make sense today. We have to identify the goals that are based upon where the science is going and where the disease is going, and of course we have to do it as partners with others, because that is really the only way we'll succeed. So in closing, I'd like to mention one other project at MVI, and I'd like to share with you a vivid memory I have of a visit with a village chief in Bankumana, Mali, probably more than a decade ago. As we were discussing with the village chief, testing vaccines in his village. And it's a discussion I had many times in the United States, and it was about this new idea or new approach of a transmission-blocking vaccine. And the idea was to vaccinate people so that they make antibodies, and when those antibodies were taken up by the mosquito, that they prevented that mosquito from transmitting malaria to others in the village. And it was crystal clear that the chief got that concept immediately. This idea of vaccinating people, not for the direct benefit, but to protect the whole village. And what he said was, we need this vaccine, we want this vaccine, and we would use this vaccine. So it's great to be part of the team here at MVI, where it's about partners, it's about the science, and it's about the future. And I do look forward to our discussion later on. Thank you.
David, very nice presentation of a lot of issues, a lot of history, and a lot of problems downstream, a lot of work to be done. If anybody has a burning question, we can take it, but I think that it'll probably, most of our discussion will be after all the speakers have, I don't see any burning, so we'll move ahead. So our next speaker is Rick Steketee, as I said before. Rick has had a distinguished career. I won't try to go through the whole history of it, but needless to say, branch chief of malaria at CDC for many, many years, and now, as I said before, science director for MSEPA, and in between, many other forays into the field and into the laboratory, distinguished career in malariology. So, Rick, look forward to hearing your comments.
Thanks very much, Carter, and thanks for everybody showing up this morning. It's nice to see some familiar faces as well as some folks that I don't know. So, and also welcome to David. It's a real pleasure to have David in the MVI team and a colleague here at PATH. So, as Carter mentioned, I've had a checkered career. I've worked in a number of different organizations. Been with PATH now and the MSEPA program since 2005, and have been working in the field of malaria. Well, depending upon when you call it, starting closing in on 30 years. So it's interesting to view the change. I get a few minutes here to talk about the successes. And one thing I would say is that, having been in the field for 30 years, or near 30 years, the speed of change is increasing. So we're dealing with acceleration here as opposed to a constant speed. And I say that because, it's been just over a decade that rollback malaria has been underway. It's arguable that in the first 5 years of rollback malaria, there was a lot of preparatory work, but not much actual change in the field. And subsequently, let's say the last 5, 6 years, it's actually been remarkable. What happened in roughly 2005 was that the Global Fund had come on board a little earlier, and by 2005, funding was actually available and moving in countries. The World Bank Booster Program started its funding effort specifically through IDA loans to countries and designating that for malaria work. And the U.S. President's Malaria Initiative began initially in 15 countries and has had a huge impact in terms of support for those countries as part of that partnership. So that meant that there were about five years or so of preparation, sometimes frustrating preparation, followed by money. And money makes a difference. It makes a difference when you actually have good science and a developed program that has clear recommendations for a set of interventions that have a scientific basis behind them. And that has led to probably the most remarkable success in child survival in my lifetime. That is a specific disease that has been categorically altered by what we've done. We've done that as a partnership. There's little question about that. And sometimes, that for those of us who have worked in this field for a long time, there are certain words that kind of dull over the years, and partnership is one of them, and integration is another, and sustainability is a third, and there are others that you hear those words and you kind of go, I'll go. But to be honest, you only understand whether or not they work when you look backwards and say, oh my God, between the Global Fund and the World Bank and the USAID and CDC and the President's Malaria Initiative and the number of other partners who have entered this field have made a difference. And the countries showed that they could deliver. So with that support, it's actually been a country ownership issue that was espoused by all of those organizations as well, that has shown that they could deliver. So people said, insecticide-treated nets in every household? How are we going to do that? Well, it's been done. Indoor residual spraying in all those targeted households? Yeah, the coverage is only in the high 80s, low 90 percentile. I mean, they can do it, okay? So this has been one of those situations where there has been a set of resources, still inadequate, but has made an enormous amount of difference. So if you walk away from anything today, let me just say that success is about people pulling together and putting money behind it and using the money well, and it's actually been done in malaria control. So we saved this past year probably about 300,000 child lives that would have died from malaria had we not been doing what we're doing. And the number, so a few numbers to try and push a couple of issues. One is that we had used the figure that there were 1 million child deaths each year from malaria around the world. And we've had that figure for a number of years. You could argue that's because we didn't actually know what the number was, but the better we get at counting that number, the more we realize that the numbers start to add up. That is, our estimates at country level across the many countries have shown that this has systematically been coming down since 2005, at the same time that population growth would have normally just driven it up. Okay, so in the last decade in sub-Saharan Africa, the population has increased about 28%. That means 28% more children available to get malaria and to potentially die from malaria if we weren't doing what we were doing. And we cut malaria deaths by 40% given the additional 28% that we had in population growth. So that's really in excess of a 50% reduction in mortality in really 5-6 years. Show me something else that gets that. Okay, measles vaccine got that. So vaccines are huge, but our package of malaria control has been really remarkable. Let me mention this, I touched on this idea of speed of change. So we're actually still in the midst of that speed of change. One example is the technologies that are available around the world that weren't there 5-10 years ago. So cell phones. So cell phones is the classic example. In sub-Saharan Africa, people didn't have phones. Well, they still don't have landlines in many places, but they now have cell phone coverage that is, absolutely remarkable. So our ability to communicate, whether it's for surveillance from health facilities around the country reporting on their cases and their severe disease and their death, or whether it's just people talking to each other, that's an example of how much change has occurred in an incredibly short period of time. And the uptake of that technology, fairly sophisticated technology, is happening at village level in what people worried wouldn't, is populations that wouldn't be able to pick this up. And the answer is they do. So, speed of change. What were we doing 10 years ago in malaria? Well, we were talking about insecticide-treated nets. We had them, and we had to put insecticide on them on an annual basis in order to keep that up. Now we have longer-lasting nets, and we're getting even longer-lasting nets, both for the net as well as for the insecticide on it. And those are in households at quite high levels. Still work to be done, but it's a remarkable difference from where we were a decade ago, where coverage was low, and that which was covered was nets that needed to be treated every year. Indoor residual spraying, an intervention that had origins back into the 40s and 50s, and stopped using it, and then started it up again as part of the package for vector control. So today we use indoor residual spraying. We had it available to us, but a decade ago, to be honest, it wasn't being used. Diagnostics. We had microscopes and slides, and a few kids got their fingers stuck and were tested for malaria, usually only if they were going to be hospitalized and needed serious treatment for their severe disease. Today, we have rapid diagnostic tests. You will hear that they're imperfect, but boy, they're so much more perfect than what we had before. It's really changed the scene in terms of how we apply or can apply our drugs. And our drugs are different. The drugs we use today were not even on the market a decade ago. So that is, this is history. But it's also about using our understanding of biology, our understanding of moving that biology forward into product development and making a difference. And so that brings me just back to the comment about the vaccine. The world has said that we can't eliminate malaria until we have new tools, like a vaccine. Well, I'd argue we've had new tools coming along almost every year, updated, improved, and what we do today is categorically different, and we really hope that what we do in the next couple of years is again categorically different. And we have that opportunity. So maybe to bring this to kind of a final point, and that is, where is this opportunity? Well, in 2007, at the Bill and Melinda Gates Malaria Forum, Bill and Melinda put back malaria elimination, eradication, that E word, back on the table. That had been taken off the table in about 1970, when there was a sense that the global eradication program for malaria started in around 1955, ending in about 1970, wasn't what we were going to do. We were going to do control. Well, the progress that we've had in this last decade have gotten us to the point of seeing control, categorically reducing severe disease infections overall, severe disease and death across huge populations, country by country. It's imperfect, but remarkably better than what it was. So when you get to that point, I'll come back to my, a word that people don't like is sustain, do we just sustain what we're doing? Well, the answer is that sustaining what you're doing when you know you could do better is actually just frustration. And we have this opportunity, having seen in a number of countries now, and MSEPA and PATH are working very closely, for example, with the Ministry of Health in Zambia and in Senegal, where there are parts of the country where there are fewer than 5 malaria infections per 1,000 population this year, in the entire year. You can actually find them, and you can go see where the infection came from and where it might have gone to, and go after those. That's what we did in this country. And actually, the Centers for Disease Control became the Centers for Disease Control because it was the malaria control program for the United States during the war. And that's what they did. They went after cases and found them and stopped the transmission locally. So we're at a point where our coverage of our interventions for vector control with indoor residual spraying and insecticide-treated nets, our ability to diagnose and properly treat the infections that we find allow us to get to places where there are so few cases we could actually stop those. In places like sub-Saharan Africa where malaria had been hugely a problem. That opportunity and not seizing it at this point would be frustrating. Maybe there are a little more serious words out there. But it does have to do with staying this course and assuring that we get the resources to make this happen. And believe me, the number of lives saved from it will be enormous. So I'll stop there and just leave you with the thought that the speed of change isn't slowing down. And the question is whether or not we are able to stay with it. Thanks very much.
Rick, thanks for that really wonderful talk about what can be done, what has been done, what can be done, and what we really need to do, we must do in the future to not only sustain, but to accelerate and come back to what Melinda Gates said at that first forum. If we keep on controlling, we'll be spending, some $10–15 billion a year forever. If we get towards this goal of elimination and eradication, then it'll be zero. So that optimistic where we go forward. So our next speaker is Hugh Chang in Seattle, where it's very early in the morning. Okay. And Hugh, I don't know as well as these gentlemen here, but he has been with PATH for quite a long time. He's an insider, if I can say that. It's my impression. And obviously into many different aspects of PATH's operations. And now taking the helm at OneWorld Health, and he's going to talk to us, I hope, about this amazing semi-synthetic artemisinin and other achievements that have taken place. Hugh?
Thank you, Carter. Can you all hear me okay? Great. Well, thank you for inviting me to speak this morning. I am honored to join this panel. I certainly don't have the experience that either David or Rick have in malaria. I'm a relative newcomer to the field. But OneWorld Health has been working on this project to create a semi-synthetic version of artemisinin for the past 8 years. And it's really, I think, a remarkable project. As you've heard from David and Rick, PATH certainly has been involved in the prevention and control of malaria. And with our work now at OneWorld Health, we have a project that can actually make a significant difference in enabling a stable supply and stable prices for the treatment for malaria. So let me just step back for a second and explain some of the background behind artemisinin. As many of you may know, artemisinin is a key component in the standard treatment for malaria called artemisinin combination therapies. The traditional route for production of artemisinin is through a plant. The plant is typically takes about 15 months from seed to harvesting and the extraction of artemisinin. And there is a dynamic that has been introduced in the marketplace because of the length of that growing time and because the effectiveness of the artemisinin that's extracted diminishes with time. So you can't grow it and store the medicines that are developed with it for a long period of time. What that results then is in these boom and bust cycles in the supply of artemisinin. Over the last 10 years, the price of the artemisinin that is provided to produce these therapies has ranged anywhere from $175 a kilogram to $1,100 a kilogram. It's fluctuated by an order of magnitude almost because of these boom and bust cycles where when farmers see that the price of artemisinin is high, they plant a lot. And then as that supply comes on the market, the prices drop because there is an excess of supply. That volatility has really affected the cost structure for the production and delivery of artemisinin combination therapies. So what OneWorld Health has done in conjunction with partners at UC Berkeley, at a biotech here in the Bay Area called Amyris, and now with Sanofi Pasteur, is to develop a process that really uses some of the latest genetic engineering techniques to basically create a non-botanical source of artemisinin. What we've been able to do is to take some early research, again, using research funds that were provided at UC Berkeley, to show that you can actually identify what the metabolic pathway is in the plant that produces artemisinin and duplicate that metabolic pathway in bacteria. The team at Berkeley had originally done this in E. coli, and we actually have been able to show, they showed the proof of concept of being able to use bacteria to produce the precursor to artemisinin, which is called artemisinic acid. With Amyris, we were able to transfer that through to a different bacterial cell line, yeast, and then with Sanofi Pasteur, we've been able to industrialize that process. And the net result of these activities has been the ability to produce at an industrial scale a non-botanical source of artemisinin that can be used to create these artemisinin combination therapies at a production level that will ultimately be equivalent to somewhere about a third, anywhere between a third and a half of the current global demand for artemisinin. The goal of the project is to stabilize supply and stabilize pricing. And what we believe we'll be able to do then through our partners is to produce by the end of 2013 about 40 to 45 tons of artemisinin and be able to supply that at anywhere between $350 and $400 per kilogram, really stabilizing that marketplace. And we believe over time, through advancements in production technologies, we'll be able to lower that cost. So I guess there are a couple of key things that I think are important about this. One is this all started with basic science. It started, I think Rick had mentioned the investments in biology, and I think David talked about how those investments in biology have helped to think about the potential for a transmission blocking vaccine that allows us to attack the parasites through antibodies that are built in the human reserves. In this case, the biology that was used was the biology of wormwood, which is the plant from which artemisinin is derived. And by understanding the basic biology of the pathway that wormwood produces artemisinin, identifying the genes that create the enzymes to enable that metabolic pathway, and transfecting bacteria with that DNA to produce those genes, we've been able to switch the supply from this plant that is grown and harvested to the bacteria that we can ferment on demand and be able to produce this new supply of artemisinin. So the U.S. support of basic research was critical to enabling this to happen at all. Partnerships, as has been in the case of all of the other discussions that we've had, are also key to this. OneWorld Health, working with UC Berkeley and Amyris, was able to provide the initial proof of concept, and then by working with Sanofi Pasteur, we've been able to develop new chemistries and new production technologies that allowed us to go from lab-scale processes to industrial-scale processes. We're very excited about the potential for this. We are, I think, by the end of this year, we will have produced a few 3 to 5 tons of initial artemisinin through the Sanofi production facilities that have been built and are beginning to become operational in Italy. And by the end of 2013, as I mentioned, we hope to be producing between 40 and 45 tons of artemisinin through this fermentation process. So we're very excited to be part of the overall PATH family. We're very excited to be able to work with our colleagues in the Malaria Vaccine Initiative and in MSEPA to be able to offer a third leg of the stool in terms of prevention, control, and treatment. And I'm happy to be able to be here this morning, early for me, to be able to talk with you.
Amazing story. It's mind-boggling. Of course, once you hear it a few times, you get used to mind-boggling things happening, and that's good. And we move on to more mind-boggling things. So we have a little time for discussion, and are there some questions? Yes.
Hi, I'm with PATH on the USAID project for child health. Hello, I'm with the USAID project on maternal and child health, MCHIP, and we were just wondering if any of the current programs you guys are running involve anemia or measuring for anemia or using anemia to judge the success of a malaria control program.
Rick, you want to take that?
Sure. Absolutely. And so, as you may realize, in terms of the large picture of anemia, there are two major groups of the population that are of concern. One is, well, maybe three. One is pregnant women who, particularly around childbirth, risk the losing blood and needing transfusions, et cetera. A second is surgical patients who need blood. And the third is young children, and there is a combination of iron deficiency anemia, and acute infections such as hookworm and malaria. Well, malaria is the one that causes the most rapid and severe drop in anemia in young children. And so when you start to control malaria, you categorically control anemia in young children, such that an example is I had an opportunity to visit a health facility in Macha, which is in Choma district in Zambia. And this is a 200-bed hospital, about 90 beds for child illnesses. When I visited there in 2005, they explained to me that in the year 2000, they had, during the malaria season, on average, somewhere between 15 and 30 children in the ward being transfused on a given day. Think about blood transfusions prior to that and HIV infection potential when blood wasn't screened as well as possible, and blood transfusions were an enormous potential for not just for helping save lives, but also for potentially giving serious diseases like HIV and hepatitis. And so today, in 2005, they had a few cases, and today they don't transfuse children. Period. End of story. So you ask about measuring that, and the challenge is that we measure anemia, and anemia has categorically changed in populations with good malaria control. The number of transfusions that we no longer give has probably actually not been quantified as well, and part of that is that blood banking systems were often run in these poor, typically rural areas where there's no blood banking. You have a child that needs a transfusion, you ask the parents to bring themselves or relatives to give blood that day in order to make that transfusion happen. And those don't get counted so easily. So we have not necessarily counted this, but I can tell you that there is story after story in health facilities of they just don't give blood transfusions to young children anymore.
Other questions? Yes, here.
My name is Fitted. I am an intern at Features Group. In areas where malaria is prevalent, HIV is also a big problem. And my question for you is, what has PATH done, especially in pregnant women living with HIV and malaria? Like, for example, I was thinking linking care of malaria with HIV. Thank you.
And as you may realize, PATH has both groups that work in malaria control, as evidenced here between MASEPA and MVI and OneWorld Health. There is a large maternal and child health group, and as part of a standard recommendation for malaria prevention in pregnancy, there is a global encouragement for women to get both insecticide-treated nets when they go to antenatal clinic, as well as to get intermittent preventive treatment for their malaria. So there is a standard recommendation in the reproductive health community for malaria prevention. That is also sitting side by side with a standard set of recommendations for prevention of maternal to child transmission of HIV. And they're delivered by the same people. So there are sets of guidelines for both of those, and that is indeed part and parcel of the reproductive health program, which is supported by PATH in many places.
So on the vaccine side, the one thing I would mention is, as part of that late stage development of RTSS, obviously a key population to look at is HIV positive individuals. So there's a study ongoing to look at both the safety and immunogenicity of RTSS in HIV positive individuals. So that's an important component of the program.
Hi, Ashley with PSI. I'm just curious, I think one of the things that we can talk about with malaria control in the last 10 years is the involvement of the private sector. And I'm just curious, as funding streams shift, vaccines are available and things, how you see the role of the private sector either shifting with vaccines, ACTs, and where you see sort of that role evolving in the next decade or so.
Quick comment. I mean, I think the goal is for it all to be in the private sector, and we don't have to be involved anymore. But with that kind of flippant answer, Rick, you want to take a shot?
Yeah. Actually, I think the private sector, and David can speak to this as well, hugely important, obviously hugely important in terms of developing new tools. At the same time that you could look at, for example, businesses in various malaria endemic countries and what their role and responsibility is for their workforce, for the families of their workforce, et cetera. And they're increasingly a set of examples of companies that have invested in malaria control for their workforce and for their families of their workforce and get return on that investment. That is, they get more money, earnings back than they spend on that. And so there is a Rollback Malaria Progress and Impact Series report on this, and if I draw your attention to that, it's out on the Rollback Malaria website. There's a site on that website on the front page that says Progress and Impact Series, and there's a private sector report on that, highlighting the fact that a number of companies, so Ashanti Gold, a company that mines gold and produces the material and sells it at huge profit, realized that when they control malaria in their workforce and get better productivity out of their workforce, they make back more money than they spend on that malaria control. Same was true for the sugar industry and for the copper mining industry in Zambia. So that's actually a good investment. It's harder to show that for the company that has 10 employees, but to be honest, in the private sector, investment in malaria control for your workforce, no matter how big or how small, is probably both a good social responsibility as well as it's likely to pay for itself, if not get you return on the investment.
Hugh, would you like to comment on that question of the role of the private sector?
Well, certainly, our goal is to enable the production of ACDs in a more economical fashion. One of the things that we have certainly experienced in our partnership with Sanofi is a very clear and open commitment to being able to supply all WHO-qualified manufacturers of ACTs with this new supply of artemisinin. So we do view, obviously, the private sector as our key constituent and the method by which this new technology will actually have its impact on malaria.
So from a vaccine point of view, it should be obvious and probably goes without saying that without GSK, RTSS would not be where it is today, and that their role in it over the last 27 years has been critical to advancing this to the point of phase 3 studies. And we anticipate that those types of partnerships are going to be critically important for next generation vaccines as well.
We're getting close to the end. We have a couple of questions. First in the front here.
Thank you very much to Dr. David, Dr. Rick, and Hugh for a wonderful presentation. I really like the word speed of change. And this speed of change, I think, has really catch up the clinician as well. I'll give an example, like in Zambia. Most doctors actually were actually considering all fever as malaria. So when malaria was actually controlled, I think they were quite confused. Now my question is, as we are controlling this malaria, I believe we have also to update the doctors. We have to update the clinician to actually do a better diagnosis, because they may actually be doing wrong diagnosis, and then we have a wrong figures of malaria being captured there. Thank you.
And was the question here, I think, or?
And I'm curious, both for the ACTs, but for the vaccine, after work with Project Optimize, so you know the issue, how much work's being done to ensure that the vaccine package produced shipped in a way that makes it easy for the ACT counterfeiting, keeping the supply chain free from corruption.
Yeah, let me take a shot at it and see, and please, you can finish off when I'm sure I'll be incomplete. But I think the gist of it is that, what about the end game of new interventions? What about the logistics? You know, what about the cold chain? What about all those things that you have a wonderful product from a company, but then how do you get it to where it's needed? And Hugh, we'll take that.
Sure, I'll start. Honestly, the work that OneWorld Health has done is upstream of the logistics. As you may know, artemisinin actually needs to go through some additional processing into one of 3 different derivatives of artemisinin that are used in the ACTs. So we will be relying on our private sector partners for the distribution of the ACTs that are developed. And OneWorld Health in particular has not focused on resolving many of those issues. I will say that through some of the efforts that are going on with respect to trying to improve access and affordability of ACTs, we're seeing spikes in the demand for ACTs. It is, I agree with you, unclear that the spikes in demand that we're seeing in terms of purchasing that seem to be going to the central stores, it's unclear how much of that is actually making it out to the districts and to the individuals who are in need of the ACTs. So I agree it's an issue. It's not one that OneWorld Health has been focused on.
I'm sure you could add some insight into this in terms of what's really happening on the ground. And
yeah, so I'll take both comments. I think the comment about the training for physicians, indeed, you know, as a physician, we're the worst. That is, we think we know better than everything else. We don't follow algorithms very well. We think we're above that. Having said that, the idea of training people, this speed of change is absolutely that. That is the health workforce that's out there, whether it's at the community level and community health workers or up into the hospitals and everything in between, is a huge issue because things have changed, and they've changed quite dramatically. On the supply chain issue, I think that you raise one of the thorns in everybody's side. It's clearly frustrating that we, the larger public health community, has been working with identifying new tools and getting, you know, the best biologic products to be available, and yet some of the systems that deliver them or should be delivering them are maybe not as strong as they should be. And this is where I would say that technology and the speed of that change potentially has us at a juncture where that could change. We now have for example, cell phones in the hands of, health workers out in rural health clinics who can call up or send a text message and say, I'm running low. And we're increasingly, in a number of places where PATH is working, are advocating for that as being part of the reporting process. That is, it's not just calling out, we had a case, but saying, we use this many diagnostic tests and we use this many ACTs and we're short of nets and this is our supply need this week. And if you don't get it to us, we'll tell you that our supply need is greater next week because we continue to use. And that should change things. And I'm raising this because I think you're absolutely right to work on it. This is a huge issue. It's not for any given disease. It's a systemic problem. And to be honest, we've had foreign assistance in this arena for decades. And it's not clear to me that we've got examples of solutions that have been made. At the same time that DHL can get you your package someplace anywhere on the globe and tell you where it is at any moment in time on that route. And so the technology is there. The incentives may not be there, and we ought to look hard at that because what's the private sector and the public sector incentive to actually deliver that service? And it has to be clear because otherwise it's not going to happen.
So maybe I'll just say a few words about the approach and the thinking that one takes in vaccines. I think we, the industry, everyone's learned kind of the hard way that some of the vaccines that have been developed initially with the developed world in mind and then have been kind of retrofitted into the developing world, that's been a tough transition. And I think, one of the standard tools that industry uses is to sit down at the beginning of the project and to map out kind of what are the key attributes that need to be made in order for this thing, one, to work, but also to be used. And it's called a targeted product profile. And to really think carefully about kind of the end user and what's going to happen. I can assure you that here at MVI and now in the malaria vaccine field, that's been a tool that's been adopted, and to start thinking about these things really early on, even at the early feasibility, so we're really clear where we're going and what those key attributes need to be. So I can tell you to change those attributes at or after the time of licensure is a huge undertaking. So, we hear you loud and clear, but I think there are some tools that can be brought to bear on this to address those questions.
Oh, just commented, coming back to the already available products. This, as you know, I'm sure, is always a very pervasive issue, and I think it's safe to say that with the PMI activities in Washington, much of the effort, and Linda can correct me if I'm wrong on this, is devoted to precisely what you're talking about. And I see emails coming across my desk that have to do with how many hundreds of units of such and such an intervention is flowing to a given place and so on at a given time. So it's huge, and from the very beginning with the target product profile right to the very end, it's huge. Yes, we have a question. Linda, you want to?
I'm Linda Banda. I work on the procurement and commodities team for PMI. So when we hear, stable supply and prices of artemisinin is something that we think about on a daily basis. I was just wondering, you had stated that the current plant-based extraction, it takes 15 months from seed to extraction. For the non-botanical process, how long is that process going to take when you're using the bacteria?
Sure, it's actually a continuous process. So there are fermentation facilities in which the bacteria are growing constantly, and we basically take batches out of those fermenters and bring them into the production line that uses the chemistry to extract the artemisinic acid, which is the precursor to artemisinin, and introduce that into the production system to convert that into artemisinin. So it is a continuous process. It is not seasonal at all. And honestly, I don't know from the beginning of the fermentation process to the output of artemisinin, I don't know for sure how long that takes, but it would be a constant supply throughout the year of artemisinin.
I think there was a question here. Yeah.
Hi, my name is Andrew Foote. I come from the water and sanitation and social entrepreneurship field. And my question has to do to continue discussing the market incentivization of the last mile of the supply chain, especially when you're talking about a vaccine. And, you know, in the water sanitation field, a term we throw around a lot is demand creation. If the panelists could share some thoughts on what have been successful demand creation stories in malaria prevention, and especially when migrating to a vaccine, what do we see as the new strategies and techniques for demand creation with the vaccine?
I think that's across the board of our panel here. So, Rick, why don't you start off?
Actually, a fair amount for the malaria vaccine. MVI, I don't mean to speak to this, David, but MVI started early on in its approach to work with countries, both at leadership level and down into communities, to ask about what the impact of a vaccine would be. Do they understand it? How do they value currently available vaccines, and what would be the differences and the issues for a malaria vaccine? So that work has already started. And it's also true across all of the interventions, and I think this is one of, going back to my comment about success, the example is that communities have taken up these interventions to high coverage when they've become available. We just had an example again in Zambia, where we've gone into a district to ask them about testing and treating the entire population of the district. And as you may know, there's a history of doing mass drug administration for populations, or in this case it was mass testing and treatment of those who were infected. People lined up in droves, and we tested probably 95% of the entire population of the district in that designated interval of time, because the message was out there that I'm going to get tested and I may have malaria and I don't know it, because many people are asymptomatic, and this would allow me to know whether or not I have it and get treated for it at the same time. And people came. And so our experience to date with all of our interventions is that this is the good side of malaria. There is not stigma around this disease. And there are no mosquito advocacy groups that I'm aware of. And so we've got a nice situation where the uptake of the whole spectrum of interventions is actually pretty darn good. Yes, there are issues, and there's some misuse issues, but this has been pretty good.
So I understand your question, actually, having come from industry and understanding demand creation and that sort of thing. What I would say, though, is sitting in MVI, I think the frame of reference is a little bit different. And what our goal is the optimal use of the vaccine, right, in a variety of different settings, and to generate the data that's required, right, and it is in a rational decision-making framework to allow the right decisions to be made in terms of how to use that vaccine. It is a new tool. How do we best use that vaccine? And so it's not really demand creation. It's how to use a new tool in the optimal way possible. And I think that's the frame of reference that we would bring to it.
Hugh, you have a comment?
Yeah, I would just echo what Rick and David have said. I would also point out that I think to the earlier question about supply and logistics systems, I think it's very important to be able to match the demand creation with the ability to actually meet that demand. And so you don't want to be generating a huge amount of demand and not be able to supply it, and then create some some dissatisfaction in those users of the various interventions. So I think it is very important, and it's very important to do it in a coordinated way across the whole spectrum of interventions and logistic systems.
Yeah, just to underline what's been said, I mean, this is very glad you pointed this out, David. There's a real difference between the interest of a public company, it's a pharmaceutical, and the community at large in terms of best use of products. And I mean, you've said it, I'll just emphasize it, that Rick has talked about how eager people are for something to help, some intervention. We don't want to take advantage of that to deliver the wrong product. So that's just to emphasize what's been saying. Yes, sir.
My question on artemisinin non-botanical production. I'm wondering whether some research done or addressing the drug resistance, whether this newly developed technology may or may not facilitate the development of drug resistance to artemisinin. Thank you.
I'm sorry, I didn't catch the end of the question.
Let me repeat it and see if I can get it right. I think I misunderstood it too, but let me give both my interpretation. So if I understand your question was, is the use of the semi-synthetic product, will that in any way facilitate development of drug resistance? On the other side of the coin, which I thought you were getting to, is there a way that you can maybe preempt the development of resistance through synthetic product? So Hugh, a couple of questions for you.
Sure. So I don't believe that the semi-synthetic version of artemisinin will have any impact, any different impact than the botanical source of artemisinin on drug resistance. Our goal, our very specific goal, is bioequivalence, so that we can reduce the regulatory hurdles that will be in place in order for this form of artemisinin to be able to be used to replace the botanical form. So we're trying to actually build the identical version, just using a different production technique. It's an interesting question about whether there may be some techniques to be able to modify the semi-synthetic version of artemisinin. I actually don't know enough about what causes drug resistance and whether, therefore, there are any changes that could be introduced that would, through this semi-synthetic process, to reduce the growing drug resistance.
David Kaslow reminded me that we have a gentleman in the room that might want to comment on that. Tom Willems.
So I'm Tom Willems, run a malaria research laboratory at the NIH, and we do quite a bit of research on drug resistance, and particularly this artemisinin resistance threat is on our radar in a big way right now. The characteristic of this threat of artemisinin resistance is in those infections. You can see it in the clinical clearance of the parasites. They're delayed. They respond slowly to the drug. And there's some evidence now that that slow clearance over the 3-day course of drug treatment will not eliminate the parasites well enough for the partner of the drug in the ACT to clear them out, and then you'll see an increased number of recrudescences coming 2 to 4 weeks later, and even longer out to 6 weeks. So the question is, can we use what we know from this basic clinical knowledge, and also from what we know of the characteristics of the drug from the plant, to develop new pharmaceuticals that can take advantage of the warhead, the active component of the artemisinin, and make it more effective in the treatment of these delayed clearances. So Hugh described the biological production in yeast of artemisinin at a synthetic level. There are also chemical synthetic methods that involve entirely new structural entities, with the peroxide warhead of the artemisinin now involving flanking chemicals, or moieties we call them, that are put together in a different way. And one of these, in particular with the Medicines for Malaria Partnership, that is a sister of MVI out of Geneva, one of these compounds called an ozonide, ozonide number 439, has a very long half-life compared to the short half-life of the drug from the plants. And it's now in advanced phase trials. It's gone through preclinical testing. And the question is, can this ozonide match the delayed clearances, or the tolerance to the herbal remedy, and be more effective, even as a one-dose therapy? So if MMV now can find a partner for the ozonide 439 that is also has suitable characteristics, it might be possible to develop a single-dose therapy with long life of the new ozonide, long life of the partner, a direct observed therapy, much like happens in tuberculosis treatment, given in the clinic, and take care of, hopefully, two problems. One is the delayed clearance of these new strains, and also the patient compliance issues over 3 days therapy. Many patients don't complete their therapy, and some studies have shown it's an abysmally low number of patients who comply with the full course, as the panel well knows. And so we have basic science, we have new drug-resistant strains to deal with, and also in delivery, the completions of the therapies and appropriate receipt of the therapy by the patients. And these new compounds are hopefully going to address many of those issues.
I see a question here.
One question I have about the vaccine. Has any thought been given to the cost of the vaccine and how it's going to be rolled out in terms of creating effective demand? Did you mention the private sector? Everything. I would not take complete issue with that, but I would wonder if we would be this far if there hadn't been substantial public support for activities like Rollback Malaria and Global Fund and PMI. So where is the effective demand entering into this in terms of how it's going to be financed once it's ready for market?
You know, I doubt that any of us here can really answer that question, but I think clearly what you alluded to is that malaria is not a high-profit, entity for entrepreneurship. So it's absolutely essential that the public sector play its role I like to think of it as an analogy to physical chemistry and to activation energy. We have to put in the activation energy into the system so that at some point, there will be an incentive for the private sector. But that's still going to come from the public. I mean, let's don't kid ourselves. The public is really going to be ultimately responsible. But over time, and I was being a little bit flippant when I said all to the private sector, if eradication is achieved, then this thing goes away. So that's a very, very long-term goal. This is eradication itself. But I think that's one of the things that was being talked about by Melinda Gates when she talked about this whole idea of resurrecting the E word. Anybody else want to comment?
Probably the only thing to point out here is that for a variety of other vaccines that are currently available in the developed world, and we're trying to provide access and availability for them in the developing world, there's what we call a dual market. So there's a private market and a public market, and there's ways to kind of balance those two to make things happen. I think one of the unique things here with this malaria vaccine, and it's going to require some innovative thinking, is how do you do that in the absence of a private market? Because this is really targeted to a public market. So I think there's going to need to be some new thinking that goes along with how do you deal with those issues. And what we've used in the past, right, for taking things from the developed world and the developing world may or may not apply here.
Let me just make a comment about Tom's mentioning of new opportunities in drugs. Just to highlight, that's yet another example of the progress that's being made, and this is a wonderful time for a balance between advances in the biology, moving to partnership in the public-private partnership for the development, and if we had a single-dose treatment, that would yet again change the name of the way things are done and the efficacy and effectiveness of programs out there. And that could happen overnight with the availability of a new drug that's a single dose, highly effective anti-malarial. So just to point out that opportunity that sits in front of us and something that we hope all comes to fruition.
Hugh, would you like to comment on this question of the role of private versus public sector going forward?
Well, I think that many of the same issues that the vaccine faces in terms of the lack of a dual market exists for many of the drugs. I think it is clear that there are many mechanisms that are being deployed to try to improve the accessibility and the affordability of these drugs, and certainly partnerships like those that have been established by MMV and those that we're developing here at OneWorld Health with pharmaceutical manufacturers to be able to reach that activation energy that David had talked about in terms of enabling an economic picture that lowers the hurdle rates for some of the private sector companies in return for achieving the public health benefits is a calculus that I think we think about in each interaction that we have with our private sector partners.
We're now really coming close to the end, but time for another one or two questions.
Thank you. My name is Mindy, and I'm from Tanzanian Embassy. I just wanted to comment a little bit on the complications, I guess, that are derived from malaria, and especially towards malaria patients. Having born and raised in Dar es Salaam, I grew up there, and just recently, my daughter and I was admitted at the hospital because she is asthmatic, so I was there for an asthmatic case. And a bed next to me was a lady with with a baby. Of course, the baby was a little bit younger than my baby. I think he was about 5 months old. And when she came back for the third time in that same hospital, that's when we were together. We were admitted in the same room, and they just couldn't find out how severe the baby's malaria was, and due to I don't know whichever complication that was, but it was very severe. And I was released the next day, but I left her at the hospital, and of course I left her with a lot of liquids and things that can help her with the baby. So 2 days later, I'm calling her just to find out, because we developed relations, and I was just trying to see how the baby was doing, and the baby had passed away. And the thing is, the problem was so real, and from talking to her, I learned that she has been to that hospital 3 times, but they said the baby didn't have malaria, and yet he had malaria, and he died from malaria. So as we're talking about advancing research and new technology, and of course vaccine for malaria, I think the disease actually for instance in Tanzanian's case, it is also evolving and developing new complications that we didn't know them before. So this genuine symptoms or the common symptoms for malaria may not exist in today's world. So as discussions are going on in eradicating malaria, and I think my government also has done quite a number of initiatives. And as a matter of fact, in Zanzibar, which is our other side of the union, malaria has been eradicated so far. So we are only battling from the mainland part of the country, whereby complications are, enormous. And as a matter of fact, you think that you have found a vaccine, and this vaccine actually is the one that I'm using for my daughter and myself. So, but again, the other side of the population, it seems like, they're actually dying from not complications that are arriving from malaria and not necessarily malaria. I don't know if it makes sense, but I just wanted you to comment on that too. Thank you.
Yeah, thanks for that. And it's, there's no question that once an individual gets infected and gets sick, a lot of things happen. And sometimes it's not just malaria, but it's other diseases on top of malaria. And because no one disease, frequently in our population, if you had a single disease, you would actually survive from that. But the problem is multiple conditions, whether it's pneumonia on top of it, or meningitis on top of it, or severe anemia, et cetera. It's complicated. I think the progress and the success that we've had in reducing the number of infections starts to mean that each one of those individual cases becomes very poignant. In addition to that, I'd point out that being hospitalized for malaria marks that individual as really at risk, not just while they're in the hospital, but when they go home, because they got their infection somewhere, and they're liable to get it again in that same environment unless we pay attention to it. So once you get the number of cases down so that it's not just overwhelming the whole population, you start to be able to address those individual cases and maybe get follow-up in the home and then prevent that repeated hospitalization, which indeed is an enormous risk. As they say, stay away from hospitals. People are dying there all the time. And so yes, you can get care there, but it is a marker that people who are hospitalized are not just at risk while they're in the hospital, but they're at risk when they go home too. So I hear your concerns, and the better we get with overall control, the better we can get with individual care.
And thank you very much for bringing this topic back to where we all want to address, to the individual child going in and not being able to be saved by the current interventions. And that's what this is all about, trying to impact on exactly what you're talking about. I think we're out of time. Thank you all very much. I think it's been a very nice discussion. I enjoyed the speakers and the comments and the questions and contributions from the audience. So let's give a hand to these three gentlemen. Thank you very much.